Thanks very much, Margaret. And you might not remember this, but the topic you gave me was vaccines for HCV and HIV, pipe dream or reality. So I'm going to put both of them in here because there are some parallels for HCV and HIV. And I guess the first question we need to ask ourselves is, why do we need a vaccine? Well, I guess the statistics speak for themselves with, in the case of HIV, 6,000 new infections per day, and for HCV, 11,000 new infections per day. And for HIV, we have a lifelong latent infection, which is integrated virus in our DNA. And for HCV, we have a chronic infection, which is different in its mechanism to HIV, but it effectively does the same thing. We can't clear the virus, but interestingly, 30% of people can clear hepatitis C virus infection. And that gives us some hope for hepatitis C that a vaccine may actually be possible. And we have fabulous treatments now for hepatitis C in the form of direct-acting antivirals that can cure more than 95% of people. And in the case of HIV, we have highly effective antiretroviral therapy, which has seen a huge decline in the number of people with AIDS, and people can now live effectively a normal lifespan with HIV. But of course, it requires lifelong treatment. The virus is not removed from the system, it remains latent. There is the possibility of drug resistance, there are comorbidities associated with long-term therapy, and only 40% of people with HIV currently receive treatment. In the case of HCV, there's a very high cost associated with DAAs, not so much in Australia, but certainly in the US. The numbers are $84,000 to $124,000 per patient. 1 to 5% of people aren't cured with the current DAAs. And that means when you've got 100 million people infected, that there might be, somewhere in the order of maybe 5 to 10 million people in the future who have drug-resistant HCV, and they will seed ongoing transmission. We also know that DAAs don't prevent people from becoming reinfected, and there is a rate of about 8 to 16 per 100 person years of reinfection for HCV. And we also have a very high number of people who are unaware of their infection status. So we also have a very poor understanding of the incidence and prevalence of Hep C because we don't do routine testing in most countries. So why don't we have vaccines for HCV or HIV? And one of the major reasons for this is that they are incredibly complex pathogens. They are remarkably simple at the genetic level, but incredibly complex for vaccine design. And one of the reasons for that is they are incredibly antigenically diverse. And in the case of HIV, the map on your left shows you just how diverse the virus is. There is 15% variation in the major target that we use for vaccines in the envelope protein within subtypes, and 25% amino acid variation between subtypes. And subtypes A, B, C, CRF01 and 2 account for 80% of HIV infections globally. In the case of HCV, we have 7 genotypes, 67 subtypes, with up to 35% amino acid variation between genotypes. And genotypes 1A and 3A account for about 70% of infections globally. So this is an enormous challenge for a vaccine to provide protection against all of these different viruses. So all of our current vaccines work because they generate what we call neutralising antibodies and sometimes cellular immune responses. Neutralising antibodies are soluble proteins that circulate in our blood. They bind to the pathogen and stop the pathogen from replicating. Sometimes these antibodies then bind to viral proteins that are on our cells, and the virus-infected cells are cleared. So we know from scientific studies that both HIV and HCV can be prevented if antibodies are administered before exposure to the virus in experimental animal studies. So this gives us hope that if we generate the right type of antibodies, we can actually prevent infection. But of course, the variation in the viral subtypes that are circulating means that the vaccines must be able to afford protection against all of the circulating strains. And one of the problems with both of these viruses, and there are a lot of parallels here, is that they are both covered in sugar groups, which are seen as self-antigens, and that makes them very poorly immunogenic. And in natural infection for both viruses, neutralising antibodies are actually delayed by up to 1 year or longer. And now I come to some specific differences between HCV and HIV. And this is a scary slide, but this is the level of detail that vaccine researchers are working at. This is the surface protein of HIV, and in the blue are the sugar groups that coat the outside of this, what we call the envelope protein. And in the boxes here are the sites of binding for neutralising antibodies. And these are all antibodies that have been isolated from infected people that have had HIV for many, many years. And there is a very small subset of people, 5 to 10%, who make broadly neutralising antibodies that are able to neutralise, that are able to neutralise a number of different subtypes of HIV. And the breadth is shown in the percentages there. Some of them, VRC01, can neutralise 95% of strains. And these are the antibodies that we want to generate with the vaccine, but they're incredibly difficult to make. So what's the problem? Well, as I said, they're very rare, and it turns out that making broadly neutralising antibodies is actually correlated with a high viral load. But having a high viral load doesn't mean you'll make broadly neutralising antibodies. It's one of those curios about HIV. The other curio about HIV is that the broadly neutralising antibodies that patients make are very unusual. They have a high degree of mutation, which is not normal of other antibodies. There's very few target sites on the envelope proteins, and they're difficult to generate. And we can't use traditional methods of making a vaccine with HIV because of safety concerns. In addition, surrogates of the envelope protein are very difficult to make. And this is where the development of an HIV vaccine has really faced a major bottleneck. For hepatitis C, I think the problem's less difficult. This is the envelope protein of hepatitis C. There are 4 major sites of binding of neutralising antibodies, and the little grey bubbly things are the sugar groups that coat the outside of the virus. And these antibodies are also very broadly neutralising. The difficulty with making an HCV vaccine, it's very difficult to grow the virus using traditional methods that we'd use to make a vaccine. And the virus also uses different mechanisms to distract the immune system from making the right type of antibodies. And it also has like an umbrella of these very strange variable regions that basically block the ability of the immune system to make neutralising antibodies. And we also have very limited knowledge of viral isolates and how neutralising antibodies are made in hepatitis C infection. If we want to contrast the two systems, HIV has benefited from billions of dollars of investment into research to understand the viral proteins and the antibody response, but Hep C hasn't seen that investment. So in terms of progress towards an HIV vaccine, the most famous of these was the RV144 trial, which was the ALVAC vaccine, an AIDS-Vax-BE vaccine, which reported a 31% efficacy versus the placebo group. This was quite a marginal difference, but it gave us hope that it would be possible to develop a vaccine for HIV. And I've just come back from the HIV meeting where we've heard a lot of progress about making an HIV vaccine using these surrogate ways of expressing the HIV surface protein. And it's now possible to start generating some cross-neutralising antibodies that give us hope that we may actually be able to generate these in humans. And those technologies are going to enter phase 1 clinical trials next year. And we also know that broadly neutralising antibodies in macaques, which is a common experimental model for HIV, can actually prevent HIV infection and prevent mother-to-child transmission. And last week, I think I heard probably 30 clinical trials at various stages that are testing HIV vaccines in humans. On the other hand, the hepatitis C vaccine story is not as happy. There's only really been one phase 1 clinical trial to generate neutralising antibodies, and it failed. and it was pulled from further development. There's currently a phase 2 clinical trial with results expected next year, which is using the other arm of the immune response, but it's likely to be very type specific. And in our lab, we've developed a experimental vaccine based on the surface protein of HCV, and we've shown that we can generate broadly neutralising antibodies. Now, I was asked to contrast this to Hep B and what we could possibly learn from the Hep B story. And the hepatitis B virus is an incredibly successful story of a vaccine. It is a very simple vaccine to produce. It's very cheap. It's made for less than $1 a dose. In Hep B, there are only 10 genotypes, and the vaccine actually protects well against most, but not all. It's 95% efficacious. It requires 3 or 4 dose schedule given to babies within 24 hours of birth, and protection lasts for up to 20 years. There's been over 1 billion doses of the Hep B vaccine given, and 184 member states vaccinate infants against hepatitis B, with 82% of children in these states receiving the vaccine. So this is incredibly successful vaccination strategy. And I guess what we can learn from that is that the vaccine that we develop, particularly for hepatitis C, should align with the hepatitis B vaccine schedule. This would be the most simple way of rolling out a hepatitis C vaccine, and in fact we've done a lot of work to develop a target product profile for Hep C vaccine based on the hepatitis B story. It needs to be very cheap because both HIV and Hep C predominantly infect people in developing countries where they can't afford expensive vaccines or complicated vaccine regimens. But the major thing that's lacking for both HIV and Hep C is advocacy. There needs to be better advocacy that we need vaccines to prevent transmission to reach elimination targets. And I don't think there's enough of that. And there's certainly not enough engagement by industry with researchers to actually co-develop vaccines. And I think for both of them, this issue of engagement with industry is really important to move this field forward. So in summary, I think for hepatitis C, a vaccine is definitely theoretically achievable, but not supported by industry. And one of the reasons for that, of course, is the DAA story. But I think we need to understand that to reach elimination targets more quickly, a vaccine is required to prevent initial infection and also reinfection. A vaccine for HIV is probably not impossible, and there's 2 parallel lines of development occurring at the moment. The recombinant vaccine antigens to generate immunity for prophylactic use, and the use of broadly neutralising antibodies as prophylactics with antiretroviral therapy. And people are looking at ways of delivering genes that generate these antibodies in people. And this circumvents the need for complicated antigen vaccine strategies that are currently stifling the field, I think. Thank you.